We are happy to welcome you at the online lecture series from the NGS-CN Summer School 2024. As most of you already know, NGS-CN stands for the Next Generation Sequencing Competence Network. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We leave the link in the footnotes, and here is a QR code for your fast access. Welcome aboard, find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Really much for inviting, and thanks everybody for joining the summer school. It's a really great summer school. So as you heard, I'm from Tübingen, from the sequencing facility in there. You just heard talk from Max, who was representing Dresden Center, and I am representing the NCCT that is located in Tübingen, and we're embedded in the university clinic. You can see a picture of it here. And the same as the Dresden and other concept genome centers, we participated in the huge DFG initiative that started for the sequencing calls, and now we're also collaborating with DFG for the individual applications for sequencing costs. So you all now heard about different sequencing platforms indeed, and that was also a really great talk. But what if you are all new to that and you would like to sequence something? So where would you start? And a sequencing project is having a certain workflow as any other project. And I would like during these 30 minutes or so guide you through the main sequencing steps in the executing a sequencing projects, also tell you what can be the problems or things to think about, and how would you generally plan something so big like that. So the usual stages for the sequencing project include planning stage, of course, one of the most important ones. Then you would need to collect the material you would eventually want to sequence. Another important step is isolating the nucleic acids of your interest, the library prep, sequencing itself, and of course, after that, that's also a very big part, which is the data processing, and you will do more and more of it in today and also tomorrow. I will not stop much on the library prep workflows, because that will be also the next talk after mine, so where you will learn it more in details. However, I will guide you through the first steps of the sequencing project. So what we usually say is that if you would divide the areas of expertise, usually that's the expertise of the lab who is coming to us with a sequencing project to envision their project and also to collect material and collect the nucleic acids. We usually help with the preparation of the samples for actually sequencing, as well provide the instruments, and we often participate in the processing of the data after that. However, what I want to stress there here is that we also can help a lot with planning a sequencing project, especially if this is something your lab does not have expertise with, because we as a facility have experience with wide range of organisms and also sample types. We also can advise, for example, on the tips and tricks for isolating your nucleic acids from problematic samples. So we would really encourage all our users of the facility to come to us still at the planning stage, because then many questions that can be problematic later on can be solved already. All right, so let's just go through the steps of the sequencing project one by one. As I said, that it all starts with the planning, and here, of course, the most important part to formulate a precise scientific question, because already the question will give you certain answers. So for example, if you are just asking what are the causes of certain pathologic phenotype that you're working on, that's a good start question. However, it's a bit too broad. What would be great that it turns into is something like which genes are differently expressed in certain organs of affected and non-affected individuals. And the question formulated like this already gives you certain answers for how exactly you want to perform your sequencing. So we already know your material type, so you will work with RNA if you're interested in gene expression. You already know the type of the sequencing you need, so obviously that will be RNA-seq. Maybe you would also want to go for the single-cell sequencing if you think that is something which will be more important. And you also can directly estimate the groups that you will compare between each other. So in this case, it will be healthy and ill individuals. And usually these kind of questions help directly to plan the mainframe of the sequencing project, and then we as a facility can help the details of how exactly it has to be executed. The next important part is to define the, so these three important steps here. So to choose the sequencing platform and the methods you're going to use, of course, to design your experiment, and last but not least is, of course, to define how many costs you have available for your sequencing project. So I know this is a bit of a busy slide, but just to guide you through what kind of questions have to be answered during this point and which questions we will ask you if you come to us with the samples for sequencing. So we would want to know which kind of platform you would go for, whether you want short reads or long reads, as Max discussed before, which kind of sequencing you would like to perform, whether that will be a bulk RNA-seq, whether you would like single cell, maybe you also need the whole genome sequencing. Sometimes we also do tailored library preparation protocols if it's something really specific. So you would need also to tell us that, whether, for example, you want a certain protocol to be executed or whether you would bring certain modifications to it. And as well, whether how exactly we should use the equipment, so the sequencing equipment, whether this is a proper simple run mode, which is something standard we use, or something very special, tailored exactly to your needs. And the another important half, which is at least equally important as the choosing of the platform and methods, is of course designing your experiment and sampling. Here, of course, you would need to decide on number of samples and also, really importantly, on number of technical and biological replicates, and of course the appropriate controls that would come with the samples you prepared. This we also can help with, because sometimes we face with the experiments that have certain controls but not the others, or for example we know that the replicate amount can be problematic later on, so we can also advise on that. Also important point that sometimes gets overlooked is the sampling strategy, especially if it's a clinical study or environmental study, how exactly samples are collected and whether they are randomized, whether we get them randomized or not, and whether they also go through anonymization and we get them already encoded. So these kind of things have also to be decided beforehand before you actually bring your samples for sequencing to us. Another important point is, of course, how much output would you like to get from the sequencing? And I think this also will be covered a bit later in the talks. So sometimes we have users that already know exactly what they want. So they would require a certain amount of gigabases or million of reads per sample, or sometimes it comes in the form of request who would like to have 100x coverage of our genome of interest. So all the things we can speak about beforehand. And also, if you don't know how much exactly you need, we have our, let's say, standard guidelines, and also we have experience in sequencing different types of the samples and different material, and we can advise you about how much output we can get or from your samples, or how much output you would require for the analysis you would like to perform later on. And of course, statistics is also important. Point when you plan a sequencing experiment, and this is, we would really advise to consult a statistician before starting the sample collections, because sometimes projects take much longer because of that, because sufficient amount of samples has to be planned and collected directly. And last but not least, it's also important to think about how the data will be transferred to you, whether we should store it, whether you would like to store it, and how it will be managed over the years to come, because all the sequencing data normally has to be made accessible and at least to a certain extent publicly available, which sometimes is not planned directly, and then it can cause extra costs as well as extra time spending on that. And of course, the costs that are available for the projects can have to balance of what would be the best option and what is possible to achieve because, of course, the reagents that we are using are very expensive, and this is unfortunately the reality of sequencing. And also, often the machines are themselves extremely expensive. That has to be also kept into account. We usually try to advise everybody on the cheapest option possible, and also to try to, for example, to suggest a bigger experiment if we know then that the price per sample will go down dramatically if we use a bigger flow cell and then, for example, provide bigger output. Sometimes we would even suggest to find collaborations if you really want to try to sequence something and have a limited budget. But bottom line here is that there is a way to arrange the optimal sequencing experiment, knowing how much costs you have available and also what are exactly the outputs you would be interested in. So after the planning phase is complete of the project, usually the material collection starts, and that's when you basically collect the samples that will later on be processed and sequenced for obtaining the data. I just would like to highlight a couple of tips for that. First of all, the best practice for material collection and storage is, of course, keeping everything stored at -80, especially that is important for RNA samples, because then that limits the degradation of the sample. And ideally, if you can snap freeze it upon collection, that's the best way to do that. We, of course, understand that it is not always an option. So this is something which is, for example, very doable in clinical settings and in the scientific lab settings. Also, the important point is that it's better not to sow your samples after they are frozen and prepared for sequencing, because this does influence the quality tremendously, especially we see that for RNA. But what I want to underline here is that sometimes samples cannot be frozen. And if, for example, these are environmental samples coming from the different parts of the world, there is no chance to directly store them properly. What we advise in this case, we advise at least to keep them as cold as you can allow yourself, or as cold as it possible in the settings you are in. And there are also some other options available for certain types of samples. For example, DNA can be dried, and that's often underestimated a lot by scientists. There is this mainly used by clinical people, the FTA cards. And actually, FTA cards provide a really great way to store samples for the transportation or for temporary storage before sequencing, and we can sequence really well material from those. So, for example, if there is no chance to cool down your DNA, you can as well just dry your DNA. And other options can be fixing the sample or lyophilizing it. They are all possible and they are to a certain extent compatible with sequencing. We just you would need to consult your facility or sequencing service provider before doing that, because some of the fixation types or some, for example, of the lyophilization protocols decrease the quality of material drastically. So we need to know about that before the material arrives to our facility, so we know there are ways to handle samples like that. Finally, RNA is usually much more sensitive to the collection and storage conditions because it's intrinsically unstable molecule, and as well because the RNases, the enzymes that digest RNA, are extremely abundant and extremely stable. That means that it is really easy to expose RNA to the enzymes that will chop it up. So in this case, if the deep freezing is not possible, using RNA preservants, like, for example, RNA Shield or RNA Later, it comes in variety of names. RNA preservants is a really good option to store your RNA in, and that also allows you to, for example, save expensive storage space in -80 and store it in just a -20 freezer. What we also advise here is just to be sure that you know what exactly storage reagent you are working with and that it is compatible with next generation sequencing workflows. Not all of them are, unfortunately, and they're also not all advertised that way. So we would also need to know beforehand which kind of preservant you used and how exactly you applied it to your samples. So another important point after the sample collection is sometimes you need to process your sample further before being able to extract your nucleic acid. So for example, if it's a human tissue, often tissue has to be homogenized before or digested with collagenase or trypsin to get the single cells, to get the cells in suspension and then expose them to the DNA or RNA extraction. Another important way to post-process your samples is, of course, cell sorting. And that's, for example, especially important for single cell applications, since you really need to have your cell suspension clear and free of cells that you don't need. So this is also something to keep in mind that can add extra costs and also extra time for processing your samples, or it sometimes can even be something as complex as the laser microdissection, which used to basically cut out pieces of tissue of interest, and then they will be processed further on for the sequencing. So basically, this is also important point that often is not considered while planning the sample collection and sample preparation, that the sample processing can also add extra time and sometimes also has to be established before you can proceed with the isolation of the nucleic acids. So after the samples are collected, the next step is actually to extract your DNA or RNA of interest. So here it's often done also by our users of the facility, but we also, for example, can offer most common workflows for DNA and RNA extraction from human tissues or from, for example, bacterial isolates, or even for certain metagenomic samples. For example, we have established workflow for soil samples or for stool samples. So what is important basically for the isolation of nucleic acids, that's to obtain sufficient amount of material to actually go into the library prep for sequencing and to obtain the sufficient quality. So I on purpose wrote here sufficient and not the best quality and best amount, because it is obvious that not all the samples will allow you to obtain best quality material. However, knowing the applications you go for and knowing the platform you go for allows you to estimate the threshold for the quality and amount of your samples. So for example, to give you an idea, if you would go for something called amplicon sequencing, so this is basically amplifying certain parts of DNA and then sequencing those parts, like the most common way would be 16S sequencing for bacterial taxonomic classification. Here we need really little amount of material. One can in principle use just 50 nanogram of DNA, and that will be completely enough. However, if you want to go for long read sequencing, there we need a lot of DNA and also in a really good quality. So that's why it's important to know what exactly you're going to, how exactly you're going to sequence your samples to be sure that you have sufficient amount of material. Another important point is the quality, of course. So here we mainly interested in 2 points in the contamination of your sample, because some chemicals inhibit greatly the library preparation for the sequencing, because library prep is enzymatic, and then some chemicals, for example, aromatic compounds, can inhibit these enzymatic steps, and then you don't get good quality library out. And another point which we are also really interested in is the integrity of the material. Especially that's important for RNA samples, because the bigger pieces of DNA or RNA we get for the library preparation, the higher quality data comes out. Obviously, for long read sequencing, this is the key point. For short read sequencing, this is also really important, because usually the workflows contain fragmentation steps. So if the material is already really fragmented, fragmenting it even more usually results in the really poor quality. But the good news here for the isolation of the nucleic acids, that there are a lot of commercial and very good commercial solutions available. That means that usually you don't have to establish everything by yourself and to reinvent the wheel. You can just check what's available on the market or check with your facility what they would use in this case. Of course, testing the DNA or RNA extraction before going for preparation of your important samples is also important. And one point that I also would like to underline is that for some samples, you might also include extra step to remove the inhibitors, that the enzymatic inhibitors that might hinder the library preparation. It's especially important for the soil samples, for example, because they have so-called humic acid. That's one of the compounds that inhibits almost all known enzymes. And for human samples, that's important usually for gut samples or stool samples, because they contain bile salts that are also inhibiting greatly the preparation of the sequencing library. I would also like to mention one important point to consider when you prepare especially human samples or samples from any other eukaryotic organisms. There is something which is called host contamination of your sample, and that depends on whether you are taking a look into the host, whether you want to know to sequence the host material, or whether you're interested in the commensal material, like for example the gut microbiota. Because what happens often with the human samples is that they will always contain certain amount of hosts, so human DNA, and it will also get processed together with the DNA of microbes or DNA of your interest, and you will sequence it as well. For some of the samples, that's not such a big problem, where you will have mainly the commensal DNA, so this is okay. You can sequence a bit of human DNA with it and remove it later on bioinformatically. But for some tissues, it's really a problem, especially, for example, for skin samples, where you would sequence 95% of the host DNA and only 5% of what you're actually interested in. So this is something which is important to keep in mind, and it already has to be encountered for during the sample collection. There are ways to mitigate this host impact, and usually they are applied to the fresh material. So that's what I meant, is that you need to plan in advance that you need to isolate your material differently. One can use differential certification analysis. There is also enrichment of microbial DNA possible. And as I mentioned, as the last resort, you can also use bioinformatic removal of the host material. But in that case, unfortunately, it might be too late and you end up with sequencing a lot of human or rodent DNA and not really the DNA you're interested in. One last point about the isolation of nucleic acids, I would like to speak a bit more about the RNA quality, because usually these are also the questions we get the most: how to obtain high quality RNA and what is a high quality RNA. So to explain here, we often use a RIN score to qualify to kind of grade the samples, the RNA samples we get. And RIN stands for RNA integrity number. And this score is ranging from 1 to 10, with 1 being the RNA of very bad quality and 10 being the RNA of really good quality. It is a bit complicated to calculate, and usually this RIN score is calculated automatically by the software, so I cannot really tell you how exactly to do it by yourself. But the bottom line here is that when we look at our RNA on the gel, the RIN score is based on the calculating the areas that are occupied by the ribosomal RNA. So this is for example, an example of very good freshly isolated RNA, and that's the migration of RNA on the agarose gel. And 18S and 20S and 28S are the ribosomal peaks. These ribosomal peaks give us an idea about how good is the RNA in general, because we can't really look at the mRNA since it's too little of it usually in the cell. However, the ribosomal RNA is a good indicator of what is in general available. And the more sharp are those peaks and the less high is the background, the better is your RNAs. For example, for in 10, they are very sharp. For in 6, you start getting a bit of degradation. So these are fragments that migrate faster. That means their RNA is generally smaller. And then it gets worse and worse until something like in 2, which is almost completely digested RNA. So this is something which we use a lot to estimate the quality, and this is something which is usually causing confusion because it's not really clear what the RIN is and how to use it. It's just a couple of general recommendations for the RNA. It's of course important to isolate your RNA in really clean conditions, ideally in the dedicated bench, since as I mentioned before, the RNases are very stable enzymes and they can survive in the lab conditions for several days. That means that they persistently can stay in your lab. It's of course important to store RNA at low temperatures, and we would recommend -80 here, and also to remember that it has to be transported accordingly also in the really low temperatures, so ideally on dry ice. Another important point which often gets overlooked is that we or the user should treat the RNA with the DNase enzyme to remove residual genomic DNA. Because for most of the isolation protocols, there will still be a bit of genomic DNA remaining after the isolation. And unfortunately, it cannot be filtered out during the library prep. So that means you'll prep the DNA as well, and then it will get sequenced with your RNA, which will contaminate, of course, your RNA reads. So this is a step which is often skipped, but I would like to underline it that it is important to at least to check whether there is a DNA contamination present in your samples. One more thing that is usually also more common for RNA is the presence of a batch effect, which basically batch effect is technical phenomenon that occurs when you process samples of different conditions separately, and then just due to the small technical difference, most probably different kit slot or maybe a bit different duration of certain incubations, your samples cluster together just basically based on the technical processing rather than on their quality. So this is, for example, a typical batch effect seen on the single cell seq, where you clearly see batches clustering together. So this is important point to keep in mind as well, to process your samples together if possible. And if not possible, please consult us in this case, and we will suggest you what is the best way to batch your samples to avoid the batch effect. And indeed, it can be mitigated later on bioinformatically. However, it's better to already include encounter for batch effect in the design of your experiment. So basically, I covered all the kind of bench parts of what you need to keep in mind for when you prepare your samples. And of course, you will do a lot of data analysis later on. I just want to underline a couple of points here that one would also need to plan while just kind of setting up your sequencing experiment. And it's not really the data analysis, which is really which has to be planned in advance, but that's something we call data management. And that is the way to make your data safely stored and also safely accessible to you after you obtain it from the sequencing. And this part is often overlooked, and sometimes we get sequencing data. So sometimes the sequencing data can be lost just because the management plan was not established beforehand. So be sure that you have a long-term storage possibility either from your university or maybe in your lab directly. The data has to be accessible by people that are working on it, and it also should be accessible in case somebody else, for example, your collaborators, have to process it. It is also extremely important to have sufficient metadata stored with your data. So that means the description of what you sequenced and what were the samples has to be available to everybody. And also, for example, the data, or at least the metadata, has to be machine readable. So it can be stored in the repository later on or can be found by other people. All right, and with that, I would like to finish this short introduction into the how to start your sequencing project. I know that it looks now a bit like this, that there are many, many factors you need to keep in mind, and all of them are important to get your project successfully done and your sequencing successfully done. But main take-home message here is that we as the facility, as the sequencing provider, can help you to answer many of those questions, especially if you don't have sequencing experience, because we already had to answer them for many other projects before, and we can give you our inputs for that. And with that, I would like to finish my talk and to wish you happy and successful sequencing.